Okay, cool. And let me share with you the today's agenda. Try to zoom in a bit. Hopefully by now we're kind of reaching steady state. So there's been a try to welcome each of you. I see. Let me check that. Yeah, not many new faces this morning. It was good to have Claudia here. Claudia was one of the pioneers that kind of got our um, got us on. I don't know. She introduced us to lots of folks that kind of increased our network, and she did some early sewing. She even sewed denim aprons for our solder folks. Um, Lots of kids these days are coming to school with ripped jeans. Yes, it's a fashion. It's not really the safest way to be soldering with open skin. So Claudia's done a number of things as well as helped us raise considerable amount of money. So I'm grateful to Claudia and happy to have her join us for a short bit this morning. Um, Otherwise, so we've got a few kind of news items we can report. I want to make sure, especially since Alicia is here, that people are able to access the private Facebook group and Instagram group. So private makes it kind of it's a double-edged sword, right? It's good to have something private, but there's also then an obstacle to joining. So I want to make sure that people are able to get in and communicate properly. Um, and then we continue to to have innovations, on the tech side, and that's a place where we can share information on our GitHub page. Um, And then the third installment is I'd like to hear from you all about um, um, your experience with the glove, good, bad, and indifferent. And then we'll hopefully maybe finish this presentation on the role of striatal dopamine in Parkinson's, especially trying to get to, the more applied aspect of, you know, how does L-DOPA work, and why does it stop working in most cases, um, and and then we can, yeah, am I in the right? Yeah, I think this is good enough. So our next meeting, yeah, will be March 22nd, and then closing insights and comments. Mm. So that's what I got planned for, excuse me, today. I haven't done enough talking yet. Um, I guess as far as just kind of news of things happening here, um, we continue to offer training for 5 teachers in northern New Mexico, um, to bring something like a glove workshop or something like a biohealth or community health workshop to their high schools, and or even middle schools, in fact. And that's our third installment was yesterday. That's funding through the Encantado Foundation out of Albuquerque. They recently held a kind of a speed networking session on Wednesday, where they invite all the local nonprofits that are funded by Encantado to begin to learn about each other and collaborate and cooperate. And I was really lucky that Emma here amongst us, and she's a senior nursing student at the college. She also went with a freshman nursing student, and also with Minna Santos, who is the spouse of our first Parkinson's patient. And together they well represented Espanola GloveWorks. They were at 3 different tables, so it seems like Espanola GloveWorks was every place. People are learning more and more about us, and so I'm grateful, doubly grateful, right, to have ambassadors out there in the community, but also not just on assignment. And I think that was transformational for some of these young people to realize, there's a lot of potential navel gazing. We just talk about how great we are. It's good to hear things from other people and to meet a bigger community that's also trying to improve quality of life in northern New Mexico. Um, And a similar thing, Santa Fe is going to hold a moving day on May 2nd. So kind of a Parkinson's outreach. and we planned a table at that, and again to get the word out on the glove. So I would invite lots of any of the local folks in northern New Mexico that might be going to the moving day on May 2nd in Santa Fe. We'll put more of that on our webpage, once we kind of lock in a table and a partner. Other other kind of local news. So the folks that are building the app have just kind of uh, have kind of continued to innovate and make I think the user interface a better experience. So for those few people that have kind of downloaded and already, we're getting some data already at the database. Um, but it's a I think once the raw stuff got kind of created, it's just gotten better and better. We did invest this week in a Mac that will allow us to develop, kind of transfer the Android app onto a Mac. And if the yeah, it's always unsafe for me to deliver exact, timing estimates for when this will happen. The base app is out there, but something that connects up with a symptom tracker and a gait tracker and a voice tracker, those components all coming together could still take us another few weeks. So, so I think that's about. One more. We have another upcoming training. We'll be training 10 teachers in Albuquerque, kind of statewide in New Mexico, on through a grant on the STEM Innovation Network in New Mexico. So again, training high school teachers on how to do kind of community health and outreach around kind of a biotech product with the glove as something principal. But there's there's lots of other kind of treatments that are health treatments that are accessible to build by high school students and teachers, not just trying to kind of stamp out a number of GloveWorks around the state. We want to give teachers some kind of skill set on how to learn from patients, and how to build devices. Um, am I missing anything else, Margaret or Minna or Emma?
I think you nailed it all,
Okay,
honestly.
okay, okay. Thanks, Emma. And then since are there, so I definitely want to move on to part three. I want to hear from folks' experience, um, and needs. But I also want to, while we have Alicia here, if there are folks that are struggling to communicate on Facebook, I want to make sure that Alicia, and I think Margaret, you're also a moderator, and probably Steve's also a moderator. Although I'm trying to delegate and uh, not have an iron grip on all facets of GloveWorks.
I
We
just
go.
sent a request, FYI.
Okay, and who's that?
Emily.
Okay, sorry. Gotcha. I've got a small gallery here since I'm hogging the screen.
Who do we go
So
for Facebook help?
say again, Ken.
Yeah, who do we go to for Facebook help?
That's Alicia Trujillo. So she's right here. Um, there's Alicia, right?
Oh,
And so she's one who set up the account. And so when you make a request, it kind of goes to her, or perhaps it might go to Margaret or myself. These are still early days.
what's the name of the group?
Espanola GloveWorks.
Okay.
I think it's the full name is Espanola GloveWorks Journal Club. No, never mind. It's just Espanola GloveWorks.
Yeah.
And at one point, right, um, Miss Janice had created an earlier account. Did she remove that, Alicia? Do you know?
I believe so.
Okay, gotcha. So that was some confusion. They're having two, but yeah. So just Espanola GloveWorks, and is it two words, Alicia?
It's one word, Espanola, then. Oh yeah. GloveWorks is one word.
Yeah, gotcha.
And they're two separate groups, then, an Instagram group and a Facebook group.
Yes, yeah, yeah.
Got it.
I've I've looked a little bit on the Facebook group. I haven't touched the Instagram yet. Um, but
And Emily, when did you send the request?
I sent it a few minutes ago, but Margaret said that she approved it.
Oh, okay, good.
Thank you.
You're welcome.
um, great. So if the social media stuff is is happening, or at least it's accessible, any other
Might be,
discussion on?
it might be useful to have a sort of a who's who with their contact information for support people for anything to do with the organization.
Yeah, that's a great. That's a great. Uh, yeah, I like that, Ken. So I'll try to put that in a number of places. Yeah,
Like if your gloves didn't show up, who you contacted.
right, right. Yeah.
Anything.
Yeah, the default address, and I'll work on this with Alicia and Margaret. Like the default address has been kind of EspanolaGloveWorks@gmail.com. And that goes to my wife Laura. And so she's really kind of the point person that can then get you in touch with the right person in the organization if she can't answer the question. Um, she's also the one that does all the shipping. Um, so. So Tom, for example, did you get your new set?
I did.
Okay,
Uh, and they're great. Really
Won-
awesome.
wonderful,
They're right
wonderful,
there, right there.
cool,
You see, it's
cool.
different, huh?
Yeah. Um,
Steve,
yes,
a question came up from two people that are using the gloves that I gave them, my modified gloves. Do they have magnets, or are they safer if you have a pacemaker?
gotcha. Got that. So I just responded to that earlier today, Ken. I'm sorry for my delay on that. I looked things up. So I mean, there are motors. Sorry, each of the motors does have a magnet. These
Okay,
are these are kind of old school motors, where there's kind of if you have if you have a moving wire in a magnetic field that kind of creates electricity or creates a motor. And so
yeah,
I wasn't able to figure out kind of the size of the motor. I mean, these are very, very small motors, of course, and so small magnets. And I don't know the specs of what size field would interrupt a pacemaker.
well, I'll just tell them they do, and I'll just tell them, because they asked the question and I just didn't know, so.
Gotcha. Yeah, they have magnets, and I sent you a link with kind of a beautiful picture of a blow up of what's inside the motor, and so you can really see the you can see the kind of geometric size of the magnet, the size of the magnetic field it generates. Um, yeah, that would be an interesting kind of subject for my students to measure the size of that field, and to kind of
I'll take a look at your email.
correlate that with what's necessary for. Um, or what need to avoid for people with pacemakers.
Thank you.
Yeah, yeah, and thanks too for sending me the photo of the rice glove. And so I do, guys. And another just brief update. I mean, our spring break at the high school is this current week, and the spring break for me at the college is the following week. So I'll be going to Houston, and I've reached out to Emmy to try to set up a meeting with her and the connections folks,
Excellent.
to kind of hopefully partner and learn more about where they're at in the process. I'll also meet with my neurology and neuroscience colleagues at UT Health Science Center at Houston to explore, kind of IRB and clinical trial research. And there's also an electrical engineer at University of Houston who's worked on something related to what Jim has kind of set up for us, right? Whether one can detect finger vibrations, in through an electroencephalogram.
Mhm.
And so there's kind of three directions happening during my week at Houston. So I'm looking forward to that. Um, any other discussion of glove issues, experiences?
There's a glove that my friend told me about that I guess just got FDA approval for essential tremors.
Mm-hmm, mm-hmm.
It's called CALAKIQ. And it's almost like a TENS pad. I think it sends electrical stimulation in the arm.
Uh-huh. Uh-huh.
But the fact that it got some FDA approvals intrigued me.
Yeah.
CALAKIQ. and then IQ are capitals. And it's CALAhealth.com.
CLAI?
No, CALA health.com.
Okay,
All
gotcha.
one word. All one word.
Yeah, essential tremor and Parkinson's seem to be, super far apart. So I just, and I haven't done enough kind of
Mhm.
research on that. But also, electrical stimulation is that's much more invasive. So it's probably even harder to get, FDA approval or something for something like that, um, as opposed to just our mechanical stimulation at the
Mhm.
fingertips. Um, yeah.
The last meeting also, I was very intrigued about that gentleman who said his brother or something was in the Synergy trial.
Yeah, and the synergy is that's the new TOS device.
They all along they're the ones who made the glove for Toss.
Yeah,
They made the one that Conor Butani had. They made him right from the start, and then they kind of parted ways. But Synergy is a company up in Oregon. But they made the gloves all along.
gotcha. Um, yeah, that's intriguing. There's lots of convergence. Things
Oh,
are going
yeah.
to happen. Things are
Oh, yeah.
happening soon, right? And if they scale up and the rice people scale up and we scale up, or even kind of combine forces as Ken's hoping Ken H. So, um,
Great.
yeah, yeah. And then may also then allow us to like collect the right data, right, to really figure out how this thing works and why it doesn't work for some folks and how to better tune it. I remain consumed by that, that as data begins to come in, and I've got more talented students that can help me kind of mine the data and kind of correlate that data with symptoms to better tune the stimulus pattern. Um, yeah. But again, that remains an aspiration. We're not there yet. So, um, but I think the app is now becoming into a state where we can collect more data. But for sure, that the bulk of our participants are kind of locked out of that because of we're yet to push it to an iPhone. So again, that will happen. Good morning, Miss Janice. We're kind of
Good
done with
morning.
an we're just we're done with announcements and we're kind of discussing experiences with the glove. And so please chime in if there's something you want us to know about.
Yeah, I was thinking, am I late? Sorry.
Um,
Or are we done?
no, we're not done. No. I've got by my clock with a change in time, it's 10:22 uh, in New Mexico right now. We started at 10.
Oh, okay. Yeah, I just wanted to check it if
Okay,
I need to know some information.
gotcha.
But I'm just a silent observer here.
Okay. Um,
So
okay. So that I can I'm here. There we go. Yeah, I was hearing some echo. Um, Yeah, so on your point, Ken, I haven't heard any more about this the second or the ongoing second, I believe, Synergy trial. We have had some folks wearing this glove that have kind of gone um, out west, for the latest clinical trial and gotten results. I don't even know how big it is. Um, some of those, well, Linda Warren is one of them, and she joins us on occasion, but I haven't heard from her in a few weeks. Um, Another was a medical doctor from Detroit whose father and uncle went out for trial and got, positive results that were also wearing, our gloves on return. So, like I said, I think it's good that there's some confluence of things happening. And it's bizarre that we're maybe not in better communication, um, maybe because all of us are still kind of under the radar or in some sense of not wondering who's got the patent and the like. So. but that's not gonna that has not slowed us down. But if there's not more discussion on kind of on the current kind of state of the glove, um, yeah, I mean, we've had a few technical innovations. The next cables that are coming will be kind of silicon insulation rather than the hard rubber. So I think there'll be a little bit more flexibility in the wires. Um, um, I did work to add kind of a low battery light, but had trouble stocking the LED. So the next versions won't go out with an LED, but the circuit's been designed. It's just a matter of kind of stocking the parts. The idea came around Chinese New Year, and so as they kind of ramp back up their production, that might change. Um, just got a new shipment of 100 more of the small computers. I'm expecting 100 more circuit boards to arrive this week. Um, our our demand is a little bit of it at an ebb right now. It's probably less than one a day of orders that we're receiving. And for me, that's kind of good. The kids are on spring break at the high school and be spring break on the college next week. So it's a little bit of a hiccup in trying to get the folks together. Um, we have
Is
brought
there any,
on we brought on a number of new interns that are kind of friends of friends have brought in more. So Friday afternoons at the high school are still buzzing with activity. But Ken H, yeah, did you have a
is there any working on, you mentioned like a silicone strap, replacing the Velcro?
yeah, yeah. Yeah, so I think on Facebook, Alicia kind of posted that new because at Amazon you can get kind of 20 of these for $6 or $7, and just kind of all the new gloves are going out with the silicone. So Tom, do your fingers have the silicone tips? Yeah, yeah, yeah.
Oh,
As opposed to the old Velcro.
The silicone works really great.
Yeah, gotcha.
It fits really tight in and they stay in place.
Mhm. Is it also then easier to put it on as well, Tom? To
I
kind of
think I think so.
okay,
The
gotcha.
whole thing seems more seamless to me.
Mhm. Um, and two, once you do size that silicone, you can kind of snip off those final tags,
Oh,
or not. Um,
that's a good idea.
yeah, just to, but it's nice that they no longer stick together, and I think they're more comfortable on the finger,
Well,
and easier to size. I think, yeah,
yeah.
um,
so is that link on the Facebook?
yeah, yeah. Maybe that's another maybe incentive. I can also put the link in the chat right now.
Appreciate that.
Yeah. Um, in fact, I might. I might put another link. So let me take 2 seconds to share with you guys something that's uh, on our main page. And so um, yeah, so can you guys see our main GloveWorks page? Am I showing that to
Yes.
you? Gotcha. Um, so I try not to make it too busy, but when I said that we're training teachers at least in the north and soon across the state to build this. And I do, and also groups in other parts of the world that want to create them. So there's this NM Mesa is that's the umbrella group of math, engineering, science achievement teachers we're working with. And GloveWorks, we now have this professional development series. So this is maybe TMI, Ken, but it's we've got 5 sessions, on how to build gloves. And the first session was, what the hell is Parkinson's? And the second one was kind of designing, printing, and wiring 4 fingers. And so here, Ken is kind of the new design of these silicone fingertips, right? And here's the Amazon link to where you can purchase those.
Okay.
And so, and what we just did on Saturday, is kind of how to print the box, how to print a circuit board, um, how to basically begin programming some of these things. Again, this is kind of TMI. This is kind of for makers, but I think Ken Toronto, for example, and a large fraction of people that first learned about the glove were makers, that were actually kind of comparing designs. And so our design is going to kind of going more and more open source, for how to pick the parts, how to stockpile the parts at the Chinese factory,
Excellent.
where to buy the computer. And so I'm hoping that this thing will be kind of self-contained for people that want to tinker, that can know how to make these things. And so one reason for these professional developments is for us to help, to build better tools so that people can do this themselves. And also, while doing it themselves, they can help us innovate, right, with maybe better ideas for things we could be doing. So I'm happy to, let me just, I will put that link, right, which again is available from our front page, but let me put that in the chat right now, unless somebody already did, because I've got so many helpers here. Okay, there's the Facebook group, and so this is the silicone straps, for lack of a better word. They're just cable ties. Cool. So there's an Amazon link now in the chat for those.
Thank you.
Okay. Great. So let me return, and we can take a few minutes to talk about dopamine and Parkinson's. So I'm going to hog the screen again, friends. Oops, that's not the right one. Great. So I'm going to go into the PDF world, and I might, yeah, there were kind of two papers under consideration. One was a large review paper, um, and I might show that one as well, right? Just to kind of jog your memory, at least picture-wise, right? So let me minimize some of the Zoom business. So this is a long review paper that's been kind of under consideration for the past maybe 2, 3, maybe 4 of our sessions. Just kind of, I've tried to highlight some of the more kind of important insights or sentences, but largely also just to kind of reinforce what's going on in the picture. And so it's about, kind of regulation of dopamine, and it's this. We all know that that in Parkinson's, the cells that create dopamine are compromised, and they vanish over time and are not replaced. But the wonders of modern science, we can put dopamine back into the system, and get it to at least give some relief temporarily, over some number of years, depending upon the individual and the time of onset, lots and lots of other variables. But I wanted to look at kind of the basic mechanism for how dopamine is, and regulation is a super big umbrella, right? So how is dopamine synthesized? How then is it released? And how then is it recycled back into the neuron for the next release? And so the top picture is kind of a very crude cartoon. It's not too much going on, but there is quite a bit going on here. So the idea is that dopamine, DA, is packaged into small vesicles. Those vesicles are trafficked to the cell membrane, at which they fuse, and to fuse membrane is then opens a pore upon which dopamine can be released to the outside world, and then activate receptors on postsynaptic cells that need dopamine in our case, to regulate voluntary motion. But there's also so many other things that dopamine is implicated in. And so there's synthesis of dopamine in the cell. It's then released, and then it's brought back into the cell by a dopamine transporter, DAT. So that transporter brings dopamine back into the cell, and the cycle continues. And as I mentioned last time, this cycle happens For the average dopamine cell in your basal ganglia, this happens 4 times a second at about more than 1 million places per cell. And so this blue cell is extruding dopamine and transporting dopamine back in at 1 million of these so-called synapses, right? These little bulges in the long kind of neurite coming out of the dopamine cell. And so the fact that they're doing this 4 times a second at 1 million places, again, makes them vulnerable to disease or attack or protein misfolding. And so that's the so-called exocytosis, release of the neurotransmitter. And endocytosis is the repackaging of the vesicles. Dopamine transport happens through the transporter. And then what we need to zoom in on here just briefly is how then, so if endocytosis is the making of these new spheres, how then does dopamine even get into those spheres? And that's something done by another transporter, but it's a vesicle transporter. These spheres are called vesicles. And so then inside the cell, dopamine is swimming around after it comes in through the transporter, and then it gets shuttled inside the sphere by a vesicular dopamine transporter. And again, everything in the world is some type of balance. You can't bring somebody in without taking somebody out because of electrical charge and just water balance and mass balance, so many other things. So these transporters are always kind of two-way beasts. And so this was how dopamine gets packaged. This is how dopamine gets moved back into the cell. This is how dopamine gets released. And I wanted to jump to the next article that's related to regulation of dopamine because it really hinges in on the role of L-DOPA, and it also talks then about also kind of L-DOPA induced dyskinesia, right? So what happens when there's too much L-DOPA in the system, when there's not enough balance? And so that second paper that I had, kind of linked for all you guys was on the so-called presynaptic effects of L-DOPA and their possible role in dyskinesia. And it's by a similar set of authors to the previous paper. So there's quite a bit of kind of reference back and forth. And I think I just, again, in the interest of, I've highlighted some of what I thought to be important sentences. Maybe this is at least one worth reading because it's a beautiful, a very elegant theory, right? And I think there's part of the elegance is it kind of resonates. It seems to make sense. So let me zoom in a little bit further there. An important attribute of dopamine and other monoamine neurotransmitter synapses is that the neurotransmitter overflows far past its release site to interact with multiple synapses, a form of volume transmission labeled social neurotransmission. And they're gonna argue that it's this kind of social, or maybe another word might be promiscuous, right? So instead of just having one-to-one contact between a presynapse and a postsynapse, dopamine is kind of dumped and then shared amongst a pool of postsynaptic cells. So that social neurotransmission seems to be essential for how L-DOPA can work. Because, there's fewer and fewer synapses there. So each synapse has to kind of do double, triple, quadruple the duty. So how could that be possible if they really were monogamous, right? If every synapse just had one postsynaptic cell, when the presynaptic cell dies, you would lose that ability to transmit. So the fact that this can be kind of shared or made more social is that a bug or a feature, right? And so it depends upon where we are in the disease. So I'm enamored of that concept. But let's again kind of jump to a picture. And sometimes pictures, at least in this state of a manuscript, are thrown to the bottom, right? So I'm gonna jump even past the references. I think there's only 2 figures in this one.
Steve,
This one,
can I ask real quick a question?
yes, please, Margaret.
So is that, and I'm sorry, I didn't read the article ahead of time, but is that promiscuity a compensatory mechanism, or is that happening even in the absence of disease?
Gotcha. Yeah, I think it happens. It happens in absence of disease. Good. That's a good question. Yeah, yeah, yeah. It happens. So it's there. It seems to be built into the. Well, it's an interesting question, right? So dopamine gets dumped into the basal ganglia for motor symptoms. It also gets dumped into the kind of prefrontal cortex as a reward signal and the like. And there in cortex, it's not promiscuous. It really is kind of one-to-one pre to post synapse. But in the basal ganglia, it does seem to really be even in the normal brain, the healthy brain. It's a bath. It's a volume dumping of dopamine. Um, yeah. And so I want to kind of maybe just go through this. I find this a very helpful kind of, healthy versus Parkinson's disease, and really what's L-DOPA doing? And so it's a little bit busy here, but there's a lot of symmetry. So let's just focus on the left side of the picture, friends, right? So this is a dopamine neuron, something where L-DOPA is translated or current turned into dopamine. And then dopamine gets packed into vesicles. Those vesicles dump dopamine out into the volume cleft and is picked up by a dopamine receptor at a postsynaptic neuron, and successful transmission has happened. Of course, once the key is turned on the postsynaptic cell, dopamine is no longer needed out there, and it can be transported back into the cell through DAT, right? The dopamine transporter. So this is this beautiful kind of recycling, right, of dopamine. So kind of synthesized, released, captured, and repackaged, and then resynthesized as well. And this is all highly, highly regulated by something called a dopamine receptor, right? And we expect dopamine receptors to be on the post side to have successful communication. But how does this cell know, right, that it's got enough or too much dopamine being dumped out? So some of this dopamine goes back to the receptor, and then this receptor regulates this mechanism. How fast will it be? How fast will new stuff be synthesized? How fast will we transport it, into the vesicle? And then how fast will we allow release? And so one mechanism for kind of compensatory mechanism, to use Margaret's word, right? So how does an amazing stat from this paper, right, is that like close to 40 or 50% of the dopamine cells have to die before motor symptoms seem to appear. So how does the system compensate for that? So during Parkinson's, these cells, you see how they get grayed out there, right? You lose dopamine cells, but you don't lose all of them. And so the key seems to be this dopamine receptor on the cell making dopamine, which again seems kind of redundant. Like, why do we need to receive dopamine on the cell making dopamine? But that's because we need a regulator. And so when these cells begin to die, the ones that are left, their dopamine receptors are noticing there's not as much dopamine out in the cleft. So the cells that remain, these receptors put them into overdrive, start synthesizing and releasing more dopamine. Also, with fewer dopamine cells out there are fewer transporters to bring stuff back into the cleft. So it seems that for the first couple of decades of Parkinson's, as these cells begin to vanish, these receptors can pick up and bring and kind of amplify the effects of dopamine, so long as they've got something to begin to synthesize, like L-DOPA that comes in artificially. Okay, but even before the L-DOPA, there's these compensatory mechanisms.
Is there any upregulation of the receptors on the postsynaptic cell in the lower?
That's a good question.
Dopamine?
That's a good question. Yeah. I don't have an answer to that. Yeah. This paper is really on the presynaptic side. That's a good question, Margaret. I don't know that, but that would make sense, right? So to get to Margaret's question, you guys, so there's the brain is so plastic, right? And so it has kind of, I think it has a target regulation, just like we have a target body temperature. And then there are lots of things inside to kind of help keep that temperature steady. And when that doesn't happen, their mechanisms kind of come into play. So with lack of dopamine in the cleft, should we then pump out more dopamine, and or should we be more receptive to the dopamine that's out there, right? And so there are these postsynaptic mechanisms and presynaptic mechanisms. And again, this is part of the marvel of the brain and the possibility of education. Nothing here is hardwired, right? And so there is this kind of chemical bath that can be modulated or regulated at the presynaptic side where dopamine gets dumped, and the postsynaptic side where dopamine gets received. But then we have to make sure we don't have the misconception that dopamine is not consumed on this side, right? Dopamine is a key that opens a gate that allows activity to happen, and then that key is released then to be resynthesized. And so if that is, and I've ignored a lot of other pathways in here just to go from L-DOPA, the drug, to dopamine, the neurotransmitter, to release, to reception, and to transport. And there's this beautiful symmetry on this picture over here. And so not only is there plasticity in the sense that these receptors can then trigger a faster rate of release, faster transport out to the cleft. Not only can we change that, but it seems that in the end stage as well, even other cells that are kind of dopamine adjacent but not dopamine. 5-HT is the chemical abbreviation for serotonin. Maybe many of you have heard of serotonin receptors. And serotonin is a neurotransmitter, or serotonin is a hormone. Right? Those are all kind of synonyms for the same thing. 5-HT just takes up less space, right? So these are normal cells that are releasing, so it's synthesizing and releasing through the same mechanism. 5-HT gets packed into a vesicle gets released. Serotonin is out there in the cleft. There are serotonin receptors on postsynaptic neurons. They are also receptors that do not consume serotonin, so serotonin is then transported back into the cell. And so, from a bird's eye view, this is exactly what's going on with dopamine, just on kind of neighboring serotonin cells. And then there's this amazing insight, right? And so not only with lack of dopamine, here we are, right? We have a grayed out dopamine cell, friends. This is one that's perished. So there's less dopamine in the cleft. Dopamine, with the help of L-DOPA, dopamine begins to get synthesized inside serotonin releasing cells. They're using the same transporter to pack vesicles. And so there's lots of evidence for dopamine overtaking serotonin in these vesicles and the release of both dopamine and serotonin. So this is just another compensatory mechanism for how important dopamine release is into the world. The world being kind of outside the cell. And again, for drug companies, this is just kind of one more pathway that they can begin to investigate. There are other dopamine machines just waiting to be used, right, in the brain if we only knew how to use them better. Right? So I find that as a positive message.
Yeah, just I don't know if anyone is on the selegiline patch. Have you guys heard of that patch? It's another drug that's used, but it inhibits an enzyme, this MAOB, that will break down dopamine. If when the dopamine is recycled back in, and before it gets put into the vesicle, monoamine oxidase will break it down. And so if you inhibit that, you actually get to, you give it time to get protected by that vesicle. And so it also helps conserve dopamine.
Gotcha. Yeah. So that's one of the pathways I was ignoring. So thank you, Margaret. Right. So so dopamine is scheduled to get packed into a vesicle, right? But then the excess can actually get degraded.
Yeah.
Again, if you can block those pathways, then you leave more dopamine available to be packaged in vesicles, allowing it to be delivered to downstream cells.
I think it ends up being like an add-on medication when
Hmm.
levodopa carbidopa is not
Gotcha. Uh-huh.
not as effective, or
So let me back up a little bit and just kind of give some context to these pictures, right? So where do they, um, again, sorry to scroll through, um, hope we're not going too fast, just to cause any seizures. Um, yeah, so maybe just I can kind of, well, yeah, not quite advanced. So there's the two places that I've focused in on here. Um, And I believe that's the case, right? So let me see if I'm really lying. Um, yeah, so maybe three, but I think I've largely stated these, but I'm probably speaking too quickly and too emotionally. So if you want a more objective scientific view, um, how does L-DOPA normalize basal ganglia function in Parkinson's? Um, these findings, which again I'm not reading, not giving you the whole paper, but it's there. Um, they beg a central issue in L-DOPA therapy. As striatal dopamine axons degenerate in Parkinson's, and only a small number of dopamine release sites remain that can be refilled, how can L-DOPA correct basal ganglia function? The answer stems in large part from properties of social neurotransmission, which we spoke about in the beginning. Um, the motor symptoms of Parkinson's appear when approximately 30% of the pigmented neurons of the substantia nigra compacta have degenerated or died. And striatal terminal density is decreased by 50 to 70%. So that terminal density, those are those little kind of blue bulges I showed you guys, right? And so losing 50 to 70% of those is when motor symptoms appear. And so that Yeah, so that's human research. It's difficult to go inside humans with part. It's not ethical to do this research. So a whole lot's been done on kind of giving Parkinson's through a number of drug regimes, giving Parkinson's to animals. So when we give Parkinson's to rats in this OHDA model, their motor function and striatal release is unchanged until about 60% of kind of DAT, right? So looking for a DAT is also one way, even in humans, to kind of to try to clinch the diagnosis of Parkinson's. So the loss of DAT is not noticed in these models until 60% of DAT is down. That's the first time that they notice motor lesions or motor symptoms. Thus, the degenerating dopamine system is able to maintain sufficient dopamine transmission to preserve motor function until relatively late stages of the disease. Right? So again, this is part of our discussion of compensatory mechanisms. This remarkable property is mostly due to two adaptive changes in presynaptic function of the spared, the remaining DA terminals, those cells, those synapses that are left that are dumping dopamine. So first, if you got diminished extracellular dopamine and reduced activation of dopamine autoreceptors, that stimulates synthesis, which may enhance dopamine release. So these are these D2 receptors that are noticing less dopamine out in the extracellular world, so they increase. And at the same time, there's just fewer cells transporting dopamine back into their cells, so there's more dopamine left for social transmission. So that's the thesis for how the brain compensates, and then how L-DOPA may help normalize. So the brain can compensate, but it still needs to producing dopamine, and so giving it some dopamine or giving it the precursor of dopamine, this L-DOPA, can allow it to kind of amp up the compensatory mechanisms. But then in parallel with that, or as a consequence of that, right, it's not all good news, in the sense that, more is not better. And as cells continue to degenerate and die, there is kind of L-DOPA induced dyskinesia. And also to connect things back to the glove, I mean, so Ken Toronto talks about, that the impact that he feels of the glove is almost like overstimulation, and dyskinesias happen is about similar to too much L-DOPA, there's such a thing as kind of too much glove or too much of the two together, um, would kind of suggest that one needs to back off either fewer drugs or ideally fewer drugs, right? Because it seems to have more side effects than the glove, right? But there are these, in kind of drug-induced dyskinesias, the topic of this discussion, right? And it's 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 a downside, right? So it's not a silver bullet. This the role of L-DOPA wears off. So by the fifth year of L-DOPA treatment, more than 50% of patients develop debilitating motor side effects, collectively defined here as L-DOPA induced dyskinesia, or LID for short. Several forms of LID have been characterized. There's a peak dose LID. There is a diphasic on-off LID. And there's an off LID, presents during unmedicated states with low L-DOPA and characterized by often painful dystonic postures. So some of you may be already, in these states, where you and your neurologist are trying to titrate the best use of this. And it's an interesting, so I might just share with you one final, more applied. Consistently, towards the end, several studies report decreased LIDs with continuous rather than intermittent exposure to L-DOPA. And and so and a recent clinical study reported that continuous delivery of L-DOPA using an intestinal gel increased on time without dyskinesia more than immediate release of the immediate release oral L-DOPA. So again, it was kind of back to Margaret's question. I'm not sure if any of you have been exposed to kind of the or studies that allow kind of intestinal gel to do the release rather than this kind of immediate release oral stuff. And this notion of kind of continuous versus intermittent, again, kind of as a wishful thinker trying to understand, what the glove is really up to, right? So is it somehow kind of is it so with the intermittent L-DOPA, right? Because L-DOPA is too strong to be on 24/7. So with kind of this with an on-off kind of ping pong, and is that kind of exposing folks to dyskinesias, you know, um, after years of use of the drug? Can we have more a continuous release of the drug through a gel, and or again, kind of this Steve kind of freelancing, is the glove giving some kind of is it extending the is it extending what L-DOPA is really trying to do, but what is it? Some folks, again, where this is coming from, more than one person wearing the glove has said it's extended their off periods, right? So I mean, in the good sense of the word, right? So their off periods, they no longer feel they know when their off period is, right? They know when the drug has stopped affecting them, but the glove seems to kind of keep the bad symptoms at bay for longer. And so again, is the glove acting again in activating this kind of compensatory network? Is it making up for the lack of dopamine? And again, and does this only work when the gloves are on, or is this kind of this kind of cumulative effect that Toss talks about? So these are questions for myself, but I'm throwing them out there. As we read things, reading science can be super super dry, but having a target question like what's going on here? So for sure, turning something on and off is really not good for the body. But also it's not super ethical to go in and implant dopamine where it's needed, right? So delivering it orally or through a gel seems to be like a natural way to go. And does the glove have a role in that kind of dopamine delivery mechanism? Or am I barking up the wrong tree? So these are trying to connect symptoms to science, right? And and so that's kind of what I had prepared for you guys to try to understand um, that paper. So Gail, please.
I have a question
Yeah.
for something. I'm an art major here.
Mhm.
So not understanding all this. But I think I understand that in Parkinson's, like 50% of the dopamine receptors are dead before there are any symptoms.
That's about right. Yes, yes.
And
Yeah.
so and some of them are being compensated for
Yes,
by the
yes.
stuff that's left.
Yeah. Yep.
Is there a point where there are so many gone that they can't be compensated for, or they can't be awakened, or whatever you might want to call that?
Yeah, yeah. So that's yeah, so that's an active field of research. Shows that there is I mean there's a point at which kind of the drug stops working. So I think late in the stage, the dopamine cells take over the serotonin cells, and that probably also has unintended consequences. Serotonin is necessary for lots of other normal functions of the brain,
Huh.
and so dopamine is so out of desperation, more and more dopamine is produced, but eventually, yeah, the L-DOPA is no longer synthesized, or it's not synthesized and delivered in a sufficient quantity
Okay.
to do anything to Parkinson's symptoms. Um, so, so the disease is progressive in the negative sense of the word, right? That things only get worse. Now, the positive upside, right, is that we do know how to grow, not we, but I mean, scientists know how to grow the dopamine cells in a dish. We know how to we know how to program a stem cell to turn itself into something that produces dopamine. And so there's literally like a Manhattan Project out there, right, of trying to understand how those cells can be implanted and made viable in the right place in the brain, right? And
And
so
if there's too many, that has bad effects too.
yeah, exactly.
So,
Yeah, yeah. So
okay.
you need to introduce something with regulation, yeah, rather than run away. Um, yeah. Um,
I wonder if that just you always hear about serotonin being implicated in depression, and I wonder if the co-occurring depression that you often see in Parkinson's happens at the point that the dopamine kind of hijacks the serotonin neurons, and
yeah.
then you get an unfortunate co-occurring serotonin depletion.
Right, right.
Hmm.
That's a great question, Margaret. Yeah, yeah.
So, Steve,
Yes, Jim.
what is the cause of these cells being killed off? Is that exposure to toxins, like in Ray Dorsey's book?
Um, yeah, that seems that's so.
I have neither of the genetic variants. I've done the test, and I don't have any of the two genetic variants that they have currently associated with Parkinson's.
Yeah, yeah,
My symptoms are caused by environmental causes, and I'm working to eliminate those. My career has exposed me to all kinds of things. So I would be a high exposure
yeah,
example.
yeah.
Now that I'm retired, I'm not doing it, but it's too late.
It's too late. Yes, yeah, yeah. So it does seem that environmental toxins are probably responsible for 70 to 90% of the cases. There's also traumatic brain injury. So,
Right.
um, but largely, it's kind of easier to avoid. Well, accidents happen, but hopefully it's easier to avoid environmental toxins. I mean, air pollution. Um, Roundup is just been kind of glorified by our current administration is something that we need for national defense, so we can deforest, other countries. Um, so and TCE, this kind of trichloroethylene,
Right.
which is brake cleaning fluid, and so
Perchloroethylene, yeah.
yeah, yeah, but it's like a lot of environmental factors, it's largely so far just kind of correlation versus causation, but the numbers are significant. And there have been some really, really bad case studies. Accidents happened near TCE spills where there was a gigantic prevalence of. So this is the Camp Lejeune story in Ray Dorsey's book.
Right.
And it does appear that these environmental insults do lead to mutations and regulation of genes that cause the misfolded proteins that would have happened if you did have a genetic. So these called so-called epigenetic. So the environmental insult affects kind of the translation and transcription of the proteins that would misfold with if they had a natural mutation, but then you can introduce unnatural mutations through environmental insults.
Right.
Like
Okay.
through Roundup or through TCE or even through air pollution.
Right.
Which is a host of particulates and the like, which is so that's something we haven't really explored, kind of the
Right.
root causes, and it's still we got there maybe a little bit in a previous discussion, right? And so there's those misfolded proteins lead to one model of Parkinson's, but there's another part of model of Parkinson's which comes from kind of degradation in mitochondria, and those seem to be targeted by other mechanisms. And I'm not sure if those are environmental or related. The mitochondrial folks are even arguing that the misfolded proteins that we think of, and we see them at the crime scene in dead cells of Parkinson's, upon autopsy, we see these tangles and we identify them as the culprits. But there's some theses that say those tangles were there, they were there as kind of a last-ditch effort to protect the cell. And so we're just seeing, we're seeing around the dead cell, we're seeing the safety mechanisms that came in to protect it. So it's
Circumstantial evidence.
yeah.
Hmm.
It's
Well,
it's fascinating and it's complicated. I'm trying to keep my my path on kind of glove related, science to try to understand
right.
how this thing can work. But
Okay.
there's amazing questions. And they're yeah, I mean, socially, they're super poignant, right? I mean, because we're poisoning ourselves, and it's the fastest growing neurological disorder, and it's within our power to limit its impact. Not for the current sufferers, but um, we need to get out the word. So
Right.
the fact that we're celebrating Roundup, when it's outlawed in most of the first world, is just it's tragic.
Right. Right. Okay.
That book, The Parkinson Plan, really points to chemicals as being the problem. Paraquat and 1,1,1 trichloroethylene. 80% of the cases they think is tied to that.
Mhm.
They're
Yeah.
not banned.
Is it more inhaled or through touch or ingestion?
All of the above.
Yeah. So I've got 11:06 friends. I don't want to keep anybody on. I do. A paper was suggested by one of our participants that I'd like to go with next time, and I might just kind of I'll push that onto the agenda, and share that with you. It's a very exciting sequence of papers that have appeared in Nature the last few years with a really beautiful kind of you know science is really good at kind of constructing kind of cartoons of how we think things work. And then other scientists come along and kind of poke holes and say, "Hey, man, that's just a cartoon. That's you don't take that literally." So I want to show you where I'd like to take us. And so I promised to give me 3 minutes just to kind of advertise. So let me grab the screen again. Yeah. And so it will also take us back to our kind of roots on trying to understand pathways in the brain. So it's a pretty bold title, Parkinson's disease as a somatic cognitive action network disorder. Um, Notice dopamine is not there in the, in the title. Um, and it has a gigantic list of international investigators from Europe, the States, and Asia. Um, let me show you what they call this somatic cognitive action network. And so hopefully this picture on the right is something that all of you have enjoyed with me over the past few months. Cross section of the brain. We're out here in cortex, and we've talked about where the foot is. We talked a lot about where the hand and the fingers are in cortex, and also, we can also take a look at the mouth because the tongue, lips are heavily innervated. But then this is just up here in cortex where kind of decisions are made about, how actually action is going to be taken and how even things are sensed from the real world. But sensing also happens through this amazing network of the thalamus, the globus pallidus, external and internal, and the putamen, the input structure of the basal ganglia, and the subthalamic nucleus, and the substantia nigra, where dopamine cells are located. So again, there's been a Manhattan Project for 60 years, on the Parkinson's network. Like, what really are the implicated places, and who talks to who governs who inhibits, who excites, and who disinhibits? These are all been fantastic questions we've been kind of immersed in. But the beautiful picture of the cortex as just, foot connect to knee, connect to elbow, connect to hand. They've found some areas in cortex now with kind of deeper studies and better imaging tools. They found some areas of cortex that were previously ignored, right, as some kind of in-between or unknown zones, so-called, even, limbic zones, limbic really meaning kind of frontier, another word for ignorance. So we're beginning to fill in gaps in cortex and see that there are some connections between these distal places in cortex and the basal ganglia. One take-home message is that it's resulted in new targets for where to do deep brain stimulation, or perhaps even where to do vibration therapy to get back to my navel gazing of like, how does the glove work? And so I'll post that paper, on our journal club page this morning. So if you want to prepare to take a look at that over the next couple of weeks, give us something to talk about on March 22nd. And with that, I wish you a very, very happy, sunny, beautiful Sunday. And
Thank you very much.
okay.
Thank you. Take
Bye,
care.
friends.
Bye.
Thank
Bye,
you.
Ken. Bye, Jen.
Hi, Steve.
Hi, Lauren.
Hi, I'm sorry. I got all confused with the time zones. We're on our way to California for a little vacation.
Gotcha.
So,
Yeah.
I'll
Yeah.
watch the recording.
Cool. Yeah, we had time to talk a lot about the science today, and a few updates as well. So yeah, I'll post the recording later today. And
Wonderful.
enjoy your holiday in California.
Thank you so
Okay.
much. See you later.
Bye.
Okay, Steve, thanks.
Thank you.
See you in a couple of weeks. Did we talk about the beach band last time?
We talked about it. Yeah, we did. Yeah.
Because that's more of a thumping. And did I mention Nick Ortner, the tapping solution?
Yes.
Okay, so
yeah,
I'm still working on trying to correlate the three of these: the gloves, the tapping, and the beach band, and see if I can draw some conclusions. I don't know what I'm doing, but something's going on with this with this manipulation of the exterior.
I agree. Yeah. So thanks for experimenting and diving in. Yeah,
I have an appointment on May 5th when I get back to the East Coast.
Uh-huh.
I have an appointment in Rochester with a movement disorder specialist, a motion disorder specialist.
Mm-hmm.
And I don't know if Ray Dorsey is still at Rochester, but that's basically where he was working. I saw a podcast of him, and he had just moved into New York City, and I was wondering what the hell is he doing there, but I'm sure he's doing something logical.
Yeah. Gotcha.
Because he's very passionate about this. So,
he is.
anyway.
He's an amazing advocate. Yeah. Yep.
So
Yeah. So,
I've read Ending Parkinson's Disease. I have to read it again. And I'm halfway through the Parkinson's plan.
Gotcha.
So
Cool.
It's like a fire hose of information
Yes,
coming at me. Right?
It is. Yeah.
Because I was just diagnosed in October, and I'm trying to figure this out.
Mm-hmm.
But there's definitely some into these gloves. Now, it's not helping my symptoms at all, but my symptoms are fairly minor. It's just my right hand that's doing the jitterbug, and I'm left-handed, so who cares?
Yes, yes.
And I can't help but think that Peter Toss's theory works better. I can't help but think that the gloves work better for mobility. There's got to be some connection there, because Parker, who was the doctor in the mid-1800s who observed his patients who came by train, which is what Peter Toss's research and gloves development is based on. It helped. I mean, maybe we diverge here for different motor symptoms. I don't know.